I think, 2 years, maybe 2.5 years.
So.
Yeah, and it has been a really, really wonderful time to have her in the lab. Some of the things about Abby, she is she is phenomenal at presenting, which you'll get to see in just a little bit. She is the type of person who will tell me that she's so excited about her work that she wants to come work on weekends and evenings, which is phenomenal. And one of the things I love about having Abby in my lab is that she is so deep into literature, and you are bringing new ideas and helping me learn new stuff about the systems that I've been learning about for years. And so I'm also very, very proud to say that Abby has been accepted to lots and lots of different grad programs and has been touring the country as she's been trying to figure out which is the best place for her. And she decided on a place. She is going to, and I don't want to get it wrong, she is going to be joining the Molecular and Systems Pharmacology program, graduate program, to get her PhD at Emory University. So congratulations. We're very proud of you. And go ahead and take us away.
Thank you, Dr. Kim. Thank you everyone for coming, and very excited today to talk about elastin-like polypeptides as versatile recombinant biomaterials for use in drug delivery. As computational power and AI have really advanced in the field of biomedicine, the number of hydrophobic drugs that is being developed is on the rise. And a lot of these are small molecule therapeutics that have benefits for a whole range of human diseases and cancers, but they are also severely limited with things like short half-lives, systemic toxicity, and poor solubility. In fact, 40% of drugs that are currently on the market suffer from low solubility. And overcoming these limitations is really important if we want the full therapeutic potential of these drugs to be realized. So to mitigate some of these concerns, we've taken a drug delivery approach in our lab. An established strategy in the field of drug delivery is to use nanoparticles. Nanoparticles work by encapsulating drugs and then conveying a lot of benefits. We see things like improved solubility and longer half-lives. Nanoparticles can also be combined with targeted delivery methods, and these can reduce systemic toxicity. To better understand how nanoparticles are really improving pharmacokinetic profiles of drugs, we can look at their composition. Nanoparticles can be made from diverse materials, things like lipids, metals, and polymers. Polymer-based strategies in particular are really important because a lot of nanoparticles are based on polymers. Polymers work by taking a monomer unit and then they are joined together by covalent bonds to form a polymer. And it's really interesting that polymers can be kind of fine-tuned in terms of their properties by adjusting things like the monomer composition and the chain length and architecture of the polymer. And these are really useful if you want to use polymers to improve drug delivery. We can kind of divide polymers into two categories. The first are synthetic polymers, and these are produced with methods of chemical synthesis, often with petroleum sources. They are associated with polydispersity and molecular weight, as well as limited biodegradability, so they can accumulate in certain areas of the body. They also can be tagged as foreign materials and then incur immune responses. The other category of polymers are natural polymers, and these are derived from materials like nucleic acids, carbohydrates, and proteins. Because they're coming from materials natural to the body, they can be very precisely controlled at the genetic level and then expressed using mechanisms like bacterial expression. They are biocompatible and biodegradable, and a lot of natural polymers are also non-mutagenic. Some natural polymers can be based on proteins, and in these cases, the monomers are amino acids linked together by peptide bonds to form a polymer that is a protein or a peptide. And if we want to use protein-based polymers in drug delivery, we have to think about the fact that there are thousands of proteins in biology, so which one do we want to pick? In our lab, we look at elastin. Elastin is a polymer that is associated with elasticity, the ability of a tissue to undergo deformation and then return back to its original shape. And we can think about parts of the body like the blood vessels or the lungs, which are contracting and expanding. We can think about the skin, which you can pinch and then see it fall back into place. All of these areas are where elastin is found. Elastin is rich in the amino acids glycine, proline, and valine, and it's cross-linked together with lysine residues. This conveys both stability and flexibility, which are very useful structural features. Because elastin is a protein that's native to the body, it does not incur an immune response, and so we classify it as non-immunogenic. Elastin is very fibrous, so these are electron microscope images of the mouse bladder, and all of those fibers that you're seeing, that's elastin. And it's very fibrous, it's very insoluble, and so this makes it difficult to work with in the lab. So what we use in our lab are elastin-like polypeptides, which are nanoparticles derived from elastin, but they have better solubility, so they're easier to work with and use in drug delivery applications. ELPs are composed of a repeating motif, where you have a valine, proline, glycine, and then X means a guest residue. It can be almost any of the amino acids, and then glycine. And this forms a pentapeptide repeating sequence that can repeat any number of times to form the ELP. ELPs are derived from elastin, which is a natural protein, so they can be produced recombinantly in bacteria. This is really useful because we can kind of harness bacteria as like living bioreactors to form lots and lots of protein that we can then use. And a key property of ELPs is that they are thermosensitive. At low temperatures, they will be soluble in aqueous solutions, but upon heating, they coacervate and precipitate out of solution. And the temperature at which this change occurs is called the transition temperature, and it is unique for every different ELP construct. You can change things like hydrophobicity of the construct or the number of pentapeptide repeats, and this will change the transition temperature, which is really useful if you're trying to think about kind of targeting an ELP for a specific application. And because ELPs are soluble at cold temperature, this makes them really useful in the clinic. Some clinical applications of ELPs will lead to a lot of benefits. These include sustained drug release, that I'll talk about more, biodegradability and biocompatibility, and again, because they're coming from elastin, ELPs are also non-immunogenic. They can be precisely encoded at the genetic level and then sustainably produced in bacteria, and they can also be combined with targeting methods to improve the way that we target drugs to specific areas of the body. One way that we allow for sustained drug release is by using drug depots. And in these systems, we can engineer an ELP sequence such that its transition temperature will happen close to body temperature. So when you're just preparing the ELPs, you can conjugate them to drugs and they will be soluble. You can put them into a syringe and then inject them into the body. But then as the temperature rises, they will become insoluble and they will form this sort of gel. And any conjugated drug that's inside the gel will be very slowly released. And so this prolongs drug release, which you can think about as being really beneficial because if you want a drug to have an effect for a longer amount of time, having sustained release really helps with that. And one way that drug depots have been used is in brachytherapy. This is a type of cancer treatment, and researchers have been able to take these radioactive seeds and conjugate them to ELPs. And then they inject them at a tumor, and what they saw was depot formation of the radioactive ELP construct. And they saw that the drug was retained at the tumor site for 2.5 times longer with depot formation compared to a construct that did not form depots. And so this was really useful for them to, in their kind of in vivo work, to see better treatment of the tumors because of the sustained drug release. So despite these uses of ELPs, they do have some limitations in terms of the way that we can incorporate them in drug delivery, and one of them is their hydrophilicity. Most ELPs are relatively hydrophilic because of the polar groups in the peptide backbone, and this limits the number of drugs that they can be compatible with. Secondly, the predominant amount of ELPs that are produced in bacteria are long, and there are potential uses for shorter ELPs that are currently untapped because of the challenges in production. And this leads me to my key question, which is to advance ELPs as recombinant biomaterials and use them to improve drug delivery. I ask two main questions, and the first relates to that issue of hydrophilicity. If we can take an ELP that may be hydrophilic, but attach it to a hydrophobic fatty acid, we should be able to form a hybrid material that can form micelles and maybe interact with a greater number of hydrophobic drugs. So what we wanted to do is see if we could improve the way that we can produce these constructs in our lab and use them to study drug encapsulation. Secondly, we wanted to address the issue of length. And if we can figure out a way to produce short ELPs recombinantly in bacteria, we can have a sustainable production method that will produce materials possibly useful in different nanoparticle applications. And I'll go into each of these questions in a little bit more detail, starting with fatty acid modified ELPs. Fatty acid modified ELPs, or FAMEs, expand the functionality of ELPs. we take a hydrophobic fatty acid and conjugate it to an ELP sequence, and this forms an amphiphilic lipid-ELP hybrid that will form micelles upon self-aggregation. In our lab, we use myristic acid as the hydrophobic fatty acid, and we can produce our FAMEs using what's called a one-pot synthesis method. This means that we take bacteria and we express our ELP gene at the same time as expressing an enzyme that will attach the myristic acid to the ELP. So we can produce the whole FAME construct using bacterial expression. We can then purify the FAMEs and they will self-aggregate to form micelles. These micelles have a hydrophilic exterior where the ELP is, and then a hydrophobic interior where the fatty acid is. And this interior can be compatible with hydrophobic drugs. If we want to study the way that they can be used in drug encapsulation, we have to improve our efficiency of production because lots of FAMEs are required to do this work. About 20 milligrams of FAMEs for each time we want to do an encapsulation experiment. So that was one of our first questions, was how can we just improve the efficiency of FAME production? And once we have FAMEs, we use them in encapsulation experiments because others have kind of laid the groundwork for this type of research. In a 2017 study, researchers took FAME micelles and used the chemotherapeutics doxorubicin and paclitaxel, and they encapsulated them in FAMEs and saw improved solubility as well as improved kinetics of drug release for these two chemo drugs. So we wanted to build on this work and see if we could expand the amount of drugs that could be compatible with FAMEs, as well as determining if we could rationally predict whether drugs will have improved solubility in a FAME system. Once we produce FAMEs in bacteria, we then need to purify them in order to use them for these studies. FAMEs are traditionally purified with a method called inverse transition cycling, or ITC, and this method takes advantage of the thermosensitive property of ELPs that I spoke about earlier. Once we have a cell pellet, we have to break open the cells and then get rid of lots of impurities before we have usable FAMEs. And so what we can do is do centrifugation steps that are alternating temperatures. At hot temperatures, the FAMEs will precipitate out of solution, and then at cold temperatures, they will resolubilize. And so by repeating this centrifugation, we can separate FAMEs from other cellular content. And this method is very well established and well documented in the literature, but it is a bottleneck in our production because it's very time intensive. Requires multiple days, so it's a lot of work that people are doing in the lab. And because there are so many repeated steps, product can be lost each time you do a centrifugation. This can really decrease yields. So we wanted to explore alternatives to ITC in our FAME production. And we looked towards a newer method called organic extraction precipitation. This method uses organic solvents, isopropanol and acetonitrile, to modulate the relative hydrophilicity of different ELP constructs. And by using these organic solvents, you can cause ELPs to either precipitate or solubilize and then be separated from other cellular debris. This method has advantages of being able to remove endotoxins. It's also much faster than ITC, so we wanted to see if we could apply this method to our FAMEs in the lab. To evaluate purification, we perform SDS-PAGE. This is an electrophoresis technique that separates proteins based on size. So anytime you see a band in the well, that represents a protein. And if you have a well that has a single band, that means that that protein is fairly pure. So looking at how SDS-PAGE works, we can now apply that to different preparations that we did of our FAME constructs. First, we can look at a gel that was taken by using ITC as the purification method. What we did was at each step of that process, every time we added a solvent or did a centrifugation step, we took a small sample and then we ran those samples on the gel. So what you're seeing is the purification in order. And you can see there are lots of bands here, so a lot of proteins are getting removed by performing ITC. And by the time we get to the last band, we have our sample, it's undergone 2 cycles of hot and cold centrifugation, and we have a single band in the well, which indicates our purified FAMEs. We can compare this to a sample that was using organic extraction precipitation. And again, we took samples at each step of that purification and ran them on the gel here. And you can see that upon adding isopropanol in that pellet band, there are a lot of bands. So there are a lot of proteins that are getting removed just by adding this organic solvent. And by the time we get to the final well, we see the presence of a single band representing our purified FAMEs. And you can also kind of see just visually by these gels that there are fewer wells in the organic extraction precipitation gel. There are fewer steps required for this much faster process. To really compare how these purification methods worked, we calculated the yield of ELP that was produced each time we did a preparation. We had 6 preparations looking at ITC as our purification method, and then 6 using organic extraction and precipitation. And we saw that with this newer method, our yields were improved 9-fold, which was a huge improvement for us. And in addition to yield, we want to think about time. So ITC requires about 2 to 3 days of work after you've done bacterial expression, but organic extraction and precipitation can happen in less than 1 day. So not only are the yields better, it's faster, this was the method that we wanted to use going forward. Once we established a good way to purify phanes, we used them in encapsulation experiments. We took phane micelles and incubated them with chemotherapeutics. And what we expect to happen is that hydrophobic drugs will partition into the hydrophobic core of the phane micelles. We did these studies on a range of drugs, and the way that we were evaluating solubility was by measuring the concentration of drug that we observed. And so that's plotted here on the y-axis, and we did this for 6 different drugs. And the pinkish red color, that's representing our systems that used phanes, comparing them to the blue color, which is a control. There were just no phanes included in these preparations. And what we saw was that solubility was improved with tazemetostat, etoposide, marinostat, panaminostat, and doxorubicin to varying degrees, as you can see based on the graph. The only drug that did not have improved solubility was decitabine, but this was expected because decitabine is relatively hydrophilic, so it's not very compatible with the hydrophobic core of our micelles. And one of the goals of this project was to see if we could predict what factors are going to influence improved solubility. And we hypothesized that hydrophobicity of a drug might determine the way that it can interact with our micelles. So we plotted solubility enhancement on the y-axis, comparing it to log P. Log P is an indicator of hydrophobicity, so a greater log P means that your drug is more hydrophobic. And you can see in this graph that there's a general trend. Increasing hydrophobicity leads to better solubility enhancement, but there are clearly outliers in this data. So we determined that log P is partially predicting solubility enhancement, but that there must be other factors at play in determining how our drugs interact with the micelles that we use. When we compared the encapsulation solvent that was used in these experiments, we looked at water and phosphine buffered saline, and we saw that encapsulation efficiency and solubility enhancement was better in water compared to PBS. And the reason why we think this happened is because when we used PBS, we saw more drug precipitating out of solution. We could just see this visually as well as experimentally. And we think that if there's less drug available in solution to partition into the micelle core, you're going to see a less dramatic increase in solubility enhancement. And that's what we think is happening in this data. Overall, we were able to determine that phanes can be efficiently produced using organic extraction and precipitation as a purification method, and that they can be used to enhance the solubility of a variety of hydrophobic drugs. What we want to do in the future is study not only encapsulation, but also drug release kinetics, and seeing if we're getting prolonged drug release by using our phane micelle systems. Maybe even observing depot formations, like those drug depots that I talked about earlier. I'll now move into the second project that I looked at, which is the issue of producing short ELPs. We wanted to develop a recombinant synthesis method to get short ELPs because we think that they could be used as surface coatings for liposomes. But what are surface coatings for liposomes anyway? I'll talk about that a little bit first. A lot of nanoparticles are coated with polymers. For example, polyethylene glycol, or PEG, is a synthetic polymer that is often used as a liposome coating. Its structure is pictured here, and in addition to its use in drug delivery, it's used in a lot of different industries. It's in skin creams, it's in toothpaste, it's in a whole bunch of things. But in terms of drug delivery, the reason why it's incorporated into liposomes is that when you add it to the exterior of a nanoparticle, it forms this hydrated shell, and this reduces recognition by the immune system and conveys what are called stealth properties to the drugs. Basically just prolonging the amount of time that that drug can circulate in the body before the immune system recognizes it. And it's used in chemotherapeutic liposomes like Doxil, which is pictured here, as well as other nanoparticle formulations. Despite these benefits of PEG, it does have a lot of dangerous consequences. I mentioned how widely used PEG is, and this means that a lot of people have pre-existing PEG antibodies, just from exposure to other PEG uses like toothpaste. So if you have these antibodies and then are prescribed a PEG-related drug, you could see allergic reactions like anaphylactic shock, even in severe cases. You can also see accelerated blood clearance, which is a mechanism that the body uses to get rid rapidly of materials that include PEG. So these negative consequences are really counteracting the whole reason why we use PEG in the first place. And if we can find a non-immunogenic replacement for PEG, this would improve the safety and efficacy of liposomes. We think that ELPs could be a potential solution. And this figure is taken from a study that researchers did where they took ELP-derived materials and they conjugated them to drugs. And using an in vivo system, they saw prolonged drug circulation by using their ELP system versus just the free drug. And so this indicates that ELP-derived materials might be able to convey stealth properties similar to PEG. And I talked earlier about how ELPs and elastin, they're non-immunogenic. So a lot of those issues associated with the immune system could be circumvented by using a more natural protein like ELPs. If we want to use ELPs as a replacement for PEG, they need to be similar in molecular weight. So the PEG in liposomes is 2 kilodaltons, so we want our ELPs to be the same size. And how can we produce 2 kilodalton ELPs? One method is solid phase peptide synthesis. This is a chemical synthesis method that can be useful, but it has a lot of kind of downsides. It can be very costly, requires harsh solvents, and it has scaling difficulties. On the other hand, synthetic biology would be a much more sustainable way of producing short ELPs. Bacteria are renewable resources. We can use lots of bacteria, produce lots of protein, and this would be a lot more efficient and sustainable. But short ELPs are very challenging to purify. Those purification methods that I talked about in the first half of the talk, those are difficult to use with short ELPs because things like thermosensitivity and hydrophobicity, when your ELP is very short, those characteristics are difficult to modulate with the purification methods that are commonly used. So to my knowledge, the shortest ELPs that have been produced recombinantly are 10 kilodaltons, much greater than the size that we need for this application. But this problem kind of has a solution hidden inside of it. What if we take a long ELP, since it's easier to purify anyway, and just produce it, purify it, and then after we do purification, then we can cut it into smaller fragments that we need. This is possible by using proteases, which are enzymes that cut proteins at very specific sequences. So what we've done is we've engineered a precursor ELP sequence pictured here. And it has that characteristic ELP pentapeptide repeat, but at very specific times in its sequence, we've incorporated the amino acid tryptophan, which is aromatic. And then we can use an enzyme called chymotrypsin because this enzyme will cleave proteins at aromatic amino acid residues. So we have a synthesis method where we will produce this precursor in bacteria, purify it, and then incubate it with chymotrypsin. And what we expect to see is that our 9 kilodalton precursor will be cut into 2 kilodalton pieces, exactly what we would need. The first step of this process is just to produce the precursor ELP. And so we express it in bacteria, and then we tested purification methods. We tried organic extraction precipitation at first, but for this construct, the yields were too low for it to be viable, so then we moved back to the ITC. And in this gel is evaluating the purification with ITC. So again, each well that you're seeing is a step of that purification process in order. And you can see a lot of bands, a lot of proteins are getting removed, and in that very last well, this sample has undergone 2 cycles of alternating hot and cold centrifugations. And we see one band at about 9 kilodaltons, which is where we would expect our purified ELP to be. After purification, we incubated the precursor with our enzyme and then evaluated the digestion process using SDS-Page. And I'll walk through just some controls first to show what we expect to see. So this first band, can you see my cursor? Yeah. This first band is the digested sample that's undergone incubation with the enzyme. And then here we have an undigested control that did not undergo incubation with the enzyme. And then finally, we ran chymotrypsin on its own on the gel just to see what it would look like and determine if it's present in other samples. And so first of all, what we see is that chymotrypsin has some molecular weight contaminants that are pretty larger molecular weights. And these contaminants are present in the digested sample as well. So we need to find a way to get rid of these if we want to go forward with this process. We can also see the precursor ELP present here in its control. That band is at about 9 kilodaltons, and this band is absent from the digested sample, which is an initial indication that that precursor ELP is being cut up into fragments by our enzymatic incubation. But there are no small peptides on this gel, even though we would expect there to be in our digested sample. So we're seeing 2 problems. First of all, there are large molecular weight contaminants that we need to find a way to get rid of. And secondly, our short ELP appears to be absent. So I'll address each of these problems in one by one to kind of go through what we did to figure out what's happening. First of all, the issue of contaminants. We decided to remove our contaminants with a method called amicon filtration. And in this method, we use a filter that has a molecular weight cutoff of 10 kilodaltons. So anything larger than that will be kind of stuck in the filter after centrifugation. But shorter materials, including our 2 kilodalton ELP, will be able to pass through the filter and then be collected, separated from those contaminants. So we performed amicon filtration and then ran the results on the gel. So we're seeing controls that I showed earlier, we're seeing samples that were retained inside the filter, and then samples that eluted through, so things that should be smaller than 10 kilodaltons. And we can look back at chymotrypsin, look at those larger molecular weight contaminants, and see that they are absent from the eluted samples, showing that amicon filtration is effective at getting rid of these impurities. But the second problem is still here. The short peptides are completely absent from this gel, even though we would expect them to be in our eluted samples. There's also a ladder band at 1.7 kilodaltons that is not on this gel either. So we were wondering, is there any protein in our digested sample at all, or is SDS page just not a good way for us to visualize that protein? So to see if there was any protein present in our digested sample, we ran a quick sample of it on high-performance liquid chromatography. And what we're measuring here on the x-axis is time of an HPLC run, and then on the y-axis we're looking at absorbance at 230 nanometers. This is where the peptide backbone absorbs, so any peak that you see in HPLC should represent the presence of a protein. We ran the undigested control, shown in pink, and then our digested sample in blue. And you can see, first of all, that there is a peak in the digested sample, so there is some sort of protein present. And the retention time is shifted. So whatever that protein is, it's different than the undigested control. To determine its identity, we ran MALDI, which is a mass spectrometry technique to determine the molecular weight of proteins very precisely. And what we saw in our MALDI data was that our short ELP had an expected molecular weight that was identical to the observed molecular weight. And this indicates that our digestion worked. We did produce 2 kilodalton ELPs with our method of enzymatic digestion. And these short ELPs, to my knowledge, are the shortest ones that have been produced with a recombinant synthetic biology approach, which is really exciting because it kind of opens up the ways that we can explore the uses of short ELPs in drug delivery. What we want to do in the future is take our precursor ELP and modify its amino acid sequence very slightly, and then complete this digestion protocol and end up with a library of different short ELPs that are all a little bit different in amino acid sequence, because this could change some of their properties slightly, and then we can take these short ELPs, conjugate them to liposomes, and test the kinetics. See if we do indeed see stealth properties like we expect. And because we have so many different ELP sequences, hopefully, this means that we can kind of optimize our results, see which one performs best at conveying stealth properties, and decide how to move forward in terms of applying these short ELPs to drug delivery. I'd like to thank Dr. Kim for her incredible work as my mentor in the lab and doing great research and always helping me do really cool things and answering my many questions. I'm very, very grateful to Dr. Kim. I'd also like to thank Jason Raymond and Elise McPherson for their work on the FAME project, as well as students in our Biochemistry 2 lab from 2024 and 2025. Graydon Holiday, he's doing great work on our short ELP side of the lab, so thank you very much for helping with that. I would also like to thank our funding sources and the Chemistry and Biochemistry department here for supporting wonderful research like Dr. Kim's lab. Thank you very much. I'd be happy to answer any questions.
All right. With the digestion, do you know what happens to the tryptophan residue?
I believe it is, if I'm remembering correctly, so the chymotrypsin works, it doesn't have like a metal, it's a non-metal enzyme, and it cuts at those aromatic amino acid residues. I think that some of the tryptophans will be incorporated into that shorter fragment that we're seeing, which is why its molecular weight is like slightly over 2 kilodaltons, like 2.2. But I think what we would want to do is like sequence all, once we have a library of short ELPs, we would want to sequence them all and like determine exactly where that tryptophan is ending up. Go ahead.
What was the guest residue composition for the FAME ELPs?
It was a GVGVP, so it was a valine.
Okay, so just FAME.
Mm-hmm.
So I was entranced by the number of bands you got for chymotrypsin.
Yeah.
Do you have a reason why that might be happening?
So the chymotrypsin that we use is on the inexpensive side, so it's probably not super pure. We did try to, we then bought a more expensive chymotrypsin that was labeled as pure, and it also had these bands. So we think that it's just something with the production of the company that we're buying the enzyme from, but because we have a way to remove them, it's okay to continue using. In an ideal world, we would use very, very pure enzyme, but here we have some impurities still present.
I have a different hypothesis.
Yeah,
So what does chymotrypsin do?
it cleaves in peptides at aromatic residues.
Right. Does chymotrypsin have any aromatic
It does.
residues in it?
So it could also be self-digestion. Yeah, I think that could be possible as well.
Yeah.
So how does, it's how you selected out, you had you've talked about it a little bit in your work, in the paper, but you said you filtered out for mass. Is that the kilodalton? I feel like there was one point where you said you got rid of everything above, was it?
In our Amicon filtration, like this
Yeah,
stuff?
that, was that
Mhm.
a mass filter or is that a size filter or?
It's a size filter. It has a molecular weight cutoff, so it will remove things that are, things that will remain in the filter will be larger than 10 kilodaltons.
So it's a size, not a
Size,
mass,
yes,
okay,
sorry,
okay,
size.
that's what I was
Yeah.
a little bit curious about. And so, and then the other thing is, the HPLC seems to be doing much more centers, it gives you a big clearer idea of what's going on.
Mm.
Why do you use, in terms of identifying, why can't you just use the HPLC as opposed to the?
To doing MALDI
Yeah.
afterwards. Once we have done MALDI once, we can confirm that what is present in the HPLC is the digested protein at the 2 kilodaltons like we would expect. But when we did the HPLC without having ever done MALDI, it's possible that what we're seeing is a different protein, some sort of impurity. Like the retention time on the HPLC isn't an indication of size. So it's important to do MALDI at least once for a particular preparation to confirm the identity of the protein. And then now going forward, if we were to repeat preparations of this particular short ELP, we could do HPLC and compare its retention time to this can now be like a standard for us. But we just had to do MALDI at first to confirm the identity of what the protein was.
So what do you think the undigested stuff is? What is, I mean, why is that showing up in such abundance there? Or what, I mean, why is it not showing in several peaks?
Yeah.
There's also a big bump after that.
So the presence of smaller peaks here, for example, that could be a remaining impurity that's still present in the sample. The reason why the peak is greater in intensity compared to the digested sample is because this was not analytical HPLC. We didn't precisely, we just kind of ran a sample. This wasn't precisely determined, we weren't kind of equating their concentrations or anything like that. So the intensity of the peak here is not as important to us. It was more just, is there a peak at all?
I'm just thinking that it's a single peak, so it's all one thing as opposed to lots of different things. And so I'm wondering if there was if that's just one. Did you guys you didn't do the MALDI on that?
We didn't do the MALDI on the undigested control. We had done sequencing on the plasmid that encodes for the gene, so we expect that it will be the precursor ELP sequence that we had designed.
First, a comment and then a question. Comment is, I don't believe it's a line in the book, but in the movie, Harry Potter and the Sorcerer's Stone, Snape says that he can teach people to bottle fame, and I note that you have bottled fame. So
That's what I was going for,
So
yeah.
first, I congratulate you on that. My question may be absurd, and if it is, you can just say, correct, your question's absurd. Do you have any interest in looking at the drugs that are actually encapsulated in your micelle systems? And if you do, are you thinking of exploring things like NLE and NMR, or putting fluorescent tags on things, or spin labels, or various ways of looking at how things are inside the micelle and how they may be interacting with the walls of that micelle?
I think that would be very interesting. It's not work that I have done, but I know some other members of the Kin Lab work with liposomes, but they will do different loading techniques and see kind of optimize loading in that way. And I think it would be interesting to apply that to ELP fans. It's not something that I've done, but I do think it would be interesting to explore.
How much is the load? Like, is it nanograms, micrograms? I mean, when you're loading a micelle, how much actually is encapsulated?
Yeah, we can calculate encapsulation efficiency, which is like a measure of once we do incubation with chemotherapeutics, how much drug are we seeing in the veins versus remaining in solution after a long incubation. And I don't have those numbers on my slides, but we did calculate that and saw that for most of the drugs, it can vary a little bit each preparation, but it is pretty efficient. There are some drugs that were better than others, I think because of maybe some of these factors in addition to hydrophobicity that could be playing a role in their interactions, but I would have to look at those numbers and get back to you in terms of what they are more precisely.
Can I ask you one other
Yes,
question?
please.
So you were talking about the depot and the gelation.
Yes.
And I think in your thesis you used the phrase timed release. Can the time release also be tuned by what you have in your ELP, or which fatty acid you're using, or do you know the answer to that?
Yeah,
I don't know if anybody does. This is
I would
what-
imagine that you could like do some fine tuning there depending on the ELP that you use, or if you conjugate like additional kind of tags to the ELP to promote different timed release. It's not work that I've done in this project, but I think it would be possible, especially if you're thinking about, I know some other members of my lab look at the brain specifically and how to cross the blood-brain barrier, and there are ways of using like focused ultrasound as a method to do that, and I think that some of those methods that are currently used with liposomes could be applied to like vein micelles as well. Yeah.
Question about neoantigenicity. So like if you add the fatty acid on the N-terminus of your ELP, you're creating something that is non-natural. So is there are there instances of adding fatty acids onto things which make something sort of immunogenic where it wasn't immunogenic before? And then the second question related to that is in natural elastins, are there forbidden guest residues? So like are there guest residues that are not naturally occurring that you can't use due to concerns about immunogenicity?
That's two great questions. I'll answer the second one first. In terms of an ELP sequence, that guest residue, it can be any amino acid except for proline. Proline would kind of distort the thermosensitivity of ELPs and make it irreversible, and we want it to be reversible so that we can do purification. So it's less to do with immunogenicity, to my knowledge, and more just about how we are producing them. And with your first question about FAMEs, I don't believe that there are instances of seeing a lot of immune responses. I know that some groups have studied FAMEs and drug formulations in mice and it did not report any adverse effects that I know of. Maybe this isn't like comprehensive. I could explore a little bit more, but at least in the FAME constructs that we're interested in, the people that are also working in this field haven't reported a lot of immune issues with our FAMEs.
Yeah, this is like a broad question, but like how are FAMEs like used commonly in like treatments of patients, or is it more like just in its infancy in terms of like being applied to medical?
Yeah, I would say it's more on the earlier side of improving development of FAMEs and expanding the number of drugs that they can work with to get into more like direct clinical trials or anything like that. Other groups have looked at FAMEs and are continuing to look at FAMEs, but it's not like, I don't think there's any like current drugs that are FDA approved that use FAMEs right now. At least I don't think so. Yeah. Go ahead.
So you mentioned that you use a specific fatty acid.
Yeah.
Is there a reason you use that specific one? Is there like a difference in the different types of fatty acids you could use?
Yeah, so we want whatever fatty acid we use, we want it to be hydrophobic because that's what's directing the micelle formation of the construct. I would imagine that you could use different hydrophobic fatty acids if you wanted to change maybe some of the compositional features of the micelles. But we've only explored myristic acid. I think it would be interesting to try it with different ones, but whatever fatty acid you use, it just has to be hydrophobic if we want micelles to form. Yeah.
What's really impressive to me is that not only are you tackling just one question, you're tackling two questions, and with sub-problems within each question, you've also had successful results in both questions. So in terms of whenever you're suggesting a alternative or a better solution to a existing thing, you can always tackle it from many different ways. Were there, like throughout your research, were there different ideas or alternatives that you tried that you thought would work, that didn't work, which ultimately led you to the choice that you made with the ones that you had the results?
Yeah, lots of things don't work. I had a lot of failure in my lab experiments. With this short ELP project, we had initially thought that the way we were doing our molecular cloning to get the precursor, that we could do some extra steps and create a longer precursor and then have better purification processes. We thought that that might be a route to explore. So I spent a lot of time doing that type of molecular cloning, and it was not working. So then we shifted to just let's just put the plasmid that we do have into bacteria. Let's just try this route, and it ended up being successful. So I had initially done some extra work that ended up not being useful. So I think that kind of thing happened to me a lot. Happens, I think, a lot generally. But yeah.
It's super impressive for the fake production. Not only were you able to get like double speed up, but also mindful yield. So that's great results both ways.
Yeah, we were looking initially just to improve efficiency time-wise, and when we saw the improvement in yield, it was like an awesome bonus. Yeah. Yeah. Okay.
Regarding the FAMEs and the organic extraction, are there any concerns about the toxicity of acetonitrile, or especially since you're or I assume this is very late stage in the drug delivery system in terms of patients you would give this to ultimately? Is there another purification step that would be necessary, I guess, in the healing?
Yeah, well, I know in terms of additional purification steps, we perform HPLC after doing organic extraction to isolate the myristylated version of our FAME and separate it from any ELP that did not undergo myristylation in case there's the efficiency of that reaction wasn't 100%. So we do HPLC there, and then after that we'll do dialysis to get rid of any HPLC associated solvents that you don't really want in your drug formulations, and we lyophilize it to get a powder of the ELP. And in terms of overall toxicity of acetonitrile, at least in the methods that we've been using with organic extraction precipitation and the literature papers that have also explored this method, it hasn't been an issue that's been commonly addressed. I think the downstream purification steps help to ensure that all that you get at the end is the thing that you want and nothing else in there.
Can I ask another question?
Yeah.
Thank you. So when you were talking about solubility enhancement, and you had a lovely slide that showed it had the log P, but then the after that it was 22C or 2C, 24C in your it just showed different solubility enhancements depending upon whether it was tasmetostat and binostat.
Yeah.
But this is two questions. One, why didn't you not, the one that you don't have here that I was surprised when looking through this, that showed real enhancement, much more so than some of these others, was this vorinostat.
Yeah.
And you guys, I didn't see that there. And then the next thing I'm gonna want you to answer is, why are these, because it's like when you make a salad dressing, you always put your emulsifier in, and you get all those flavors into the center, and we never think about why some things get into the middle and some don't. But in the same thing here, sometimes some of these drugs are very successful, others are not, but they're all trying to go towards the same environment. It would seem that they would all want to be there, and why do you think there is some discrepancy?
Yeah, we had also kind of thought initially that hydrophobicity would be like the leading determination of how drugs would interact into getting into that core of the micelle. But upon seeing some of these outliers like panobinostat, which is less hydrophobic than TAZ but has better solubility enhancement, so we were thinking that there could be other structural factors in terms of the drugs, maybe like geometric things like stacking of aromatic rings, or like, other steric factors. Maybe there's just other interactions in terms of how these drugs are, when you're using a lot of them in an experiment, how they're going to interact with the micelles. So we would need to do more data to like confirm those factors, but we were hypothesizing that some of those might be at play in addition to just hydrophobicity. Does that answer your
Yeah.
Okay.
So why didn't you do the VOR
For the
in the water
For the
versus-
water solubility,
Yeah.
yeah. We just took a subset of the drugs that had better solubility to test the encapsulation solvent, just to like see if this parameter would have an effect at all. And now seeing that it does have an effect, we would ideally like to go back and repeat this with all of the drugs. But we had just done a subset to determine if it had an effect at all.
So the answer is that you have to graduate and you don't have enough time.
That is the answer, yeah.
Right.
And related to that, I guess, have you plotted this with the encapsulation efficiency as
Mhm.
well? Is there any other correlation that you see between those?
Not a super strong correlation. So I think that there would be those other factors might be more predicting the encapsulation efficiency versus just hydrophobicity.
Has anybody computationally studied what one of these things passing through my cell to get encapsulated, what the dynamics of that might look like? I know you haven't done it, I know you're not going to do it, but has anybody?
In the
looked at-
literature?
Yeah, has anybody looked at that?
I'm sure
Because
that
I'm
there.
thinking the big deal, of course, in chemistry is always thermodynamics
Yeah.
versus kinetics.
Yeah.
And we need to think about the thermodynamics of the interactions, which is what you're talking about, hydrophobicity and all of that. But there's also, of course, the kinetic question of how do these things migrate through your systems. And I was just wondering if anybody's looked at that kind of thing.
Yeah, I haven't come across a lot of computational work related to fame micelles. I think like studying micelles generally can be a little bit of a broader field, but I haven't personally come across research in that related to fames.
Thanks.
Thanks for it, Gabby. I'm curious, I'm really fascinated by this temperature sensitivity.
Yeah.
And I'm curious, in the overexpression, have you tried, or has anybody tried different growth temperatures for the coli?
Yeah,
to,
yeah.
To
That
increase
can-
potentially yield?
That can definitely be manipulated, and something that like we've explored in our lab is temperature related to the length of incubation, because like, at some point it's like helpful to incubate for a long time. You get a lot of protein in the bacteria, but then at too long you kind of can see a plateau.
Yeah.
And so people have kind of looked at those parameters, and we've tested it very practically in our lab in terms of like, when you're working on research during the semester, it's like, how long are you gonna leave something on the incubator? So we've done different ways kind of inadvertently that way, and we've kind of seen, like, we do ours at 37 degrees Celsius for 18 hours, and that works really well to get protein expression to a level that is kind of right at that sweet spot of having a lot of protein without hitting that plateau.
And then the depots, I know you haven't done this or and aren't going to do it, but how would you visualize that? You said like one of the future things would be to be looking at these depots. How is that done?
Yeah, we would first look at drug release kinetics, like the amount of time that a drug formulation is circulating in the body, and you can study that with like the concentration of drug in the plasma or something like that. And usually a longer drug circulation would be an indication of depot formation if those drugs are very slowly leaching out. I think that you could also do some like size-based techniques and see if you're, I don't know exactly how this would work, but I would imagine you could see, like, I know people have done it with liposomes, but like seeing like size of a liposome, like, depot kind of clump would be bigger than just liposomes with drugs in the body, and I think we could maybe apply techniques like that, study that to ELP micelles. But usually drug circulation and drug release is a good indication of depot formation.
That's
rather, not really a direct measurement.
Yeah,
of
more,
the
yeah,
visualization of the
indirect.
depot.
Yeah, exactly.
Okay. That makes sense.
So the migration just to get out of those depots is just, that's just slowly they're gonna leak out.
Yeah, slowly
And
they'll
that's-
leak out, especially if you have like a big aggregate of ELPs, drugs in the middle, like will just take longer to get outside of that aggregate. Especially if they're like forming kind of insoluble gels.
So if you may, okay, because the gel is, if it's, they'll break down eventually. and
Yes,
float
it just
loose
takes
and go,
a
okay.
while, yeah.
You kind of mentioned towards the beginning that in addition to like the solubility enhancement and the slower drug release, the protective effects being on like encapsulated in myself. Did you notice that at all for some of the more rapidly degrading chemotherapeutics, that there was a benefit?
That's work that people have explored a little bit. That initial study that I pointed out with doxorubicin and paclitaxel, those researchers did see prolonged circulation in the body with using a FAME, or not the body in their like mice system, but in their FAME system. And we, like our work here has been encapsulation focused, so we would want to move into the kinetic studies and see if there is that kind of like protective benefit there.
All right, well, if there are no more questions, let's give Abby another round of applause. Well, thank you all for showing up. I believe the chemistry faculty will now have to come together and deliberate on the defense.
Thank you for coming.